Okay, so today we're going to be talking about the second half of chapter 22. You've already done first half of chapter 22, so let's go ahead and jump straight into this. So we're going to start with the adaptive immunity, and let me go ahead and share this image here. Okay. Okay, that's not really going to work for what I want to do. Oh, okay. All right, but this is where I'm going to pick up, and this image shows you the kind of a breakdown of the adaptive immunity. So what is what, and of course we're going to be talking about way more details than this. This is a good figure to be familiar with. Okay, so hopefully you can maybe redraw this figure and see if you can kind of see where things fit. Okay, so adaptive immunity is divided into two types of immunity. You have the cell-mediated immunity, and this is going to involve your T lymphocytes. And then you have the humoral immunity, and that's going to involve your B lymphocytes. So that's a very important thing to be aware of. Okay, so cell-mediated immunity, a part of the adaptive immunity is dealing with T lymphocytes. And then humoral has to do with the fact that it's going to involve antibodies also, which are going to be part of your blood plasma, and that's why it's called humoral. But it involves the B lymphocytes that produce antibodies. So under the cell-mediated immunity, you can see that when we talk about the T lymphocytes, we will be talking about helper T lymphocytes and cytotoxic T lymphocytes. And then for the humoral immunity, we'll be talking about the B lymphocytes, which will eventually become plasma cells. And these are going to be the ones that produce the antibodies. The effector response, that terminology simply means what is the effect of that type of immunity. So effector response of your T lymphocytes or cell-mediated immunity would be for the cytotoxic T lymphocytes, it would be destruction of the infected cells. For helper T lymphocytes, it would be activation of other immune cells. And then for effector response of the humoral immunity, this is the antibody production. So antibodies in the blood plasma. Okay, so that's kind of our breakdown here. So the adaptive immunity is going to involve antigens and antibodies. Okay. So antigens, so adaptive immunity is initiated by this contact with antigen, and antigen can be, generally it's a foreign substance, and I saw that actually whenever I was grading your blood labs, I saw that a lot of people were saying antigen is a foreign body, which in a lot of cases it is, but we also have self-antigens. So in the case of blood typing, the antigens, if we're talking about someone that, that person's blood type, the antigens are actually not foreign. They're on your own erythrocytes, so they're your own. But in general, the antigens that are involved in the immune response are going to be foreign bodies. So something that's not originating in your body. And they're going to be specialized lymphocytes that will form against these antigens. And adaptive immunity, adaptive response is going to take several days. And this should already make a little bit of sense if you have watched the first part of our immune system, because you know that for example, if there's inflammation going on, right, that process is basically leading into activation of the adaptive immunity. Uh-oh. One person that I didn't know was in the waiting room. Sorry about that. So that's that. So it takes several days. It's not as quick as your first line of defense or your second line of defense, okay? Because the first and second line of defense will ultimately aid in activation of this adaptive immunity. I've already mentioned that we have cell-mediated immunity and humoral immunity. So just remember, cell-mediated, it's T lymphocytes, and then humoral immunity is your B lymphocytes producing antibodies. So it's really the antibodies that are involved there. So let's talk a little bit more about the antigens. Antigens are molecules or substances really that are going to bind antibody or a T cell. It could be a cytotoxic or helper T cell, and depending on which one it is, there's going to be different things that will happen. So the antigen molecule can be, it's usually a protein, but it could be a large polysaccharide, especially like if we're talking about bacteria, and they tend to have things hanging off of their capsules. So it could be that, but a lot of times it's a protein. A little bit of a terminology that I need you to be aware of, and that's just, knowing your vocabulary here. Okay, so make sure that you are aware what the antigenic determinant means, or epitope. It's the same thing, but this is the site that is recognized by the immune system. So this is where this is part of that antigen that your immune system is going to attach to. Immunogen is something that it's an antigen that causes an immune response. So in general, like your self antigens should not be antigens that cause an immune response. Okay, self antigens should not cause immune response in your own body, and therefore should not be immunogens. Okay, so immunogen is something that triggers your immune system, that stimulates the immune system and causes your body to go into that immune response. And then another word that I want you to be aware of is haptens. Haptens are molecules that are too small to act as antigens on their own. So they're going to bind another molecule, and after they do this, that's when they will trigger immune response. So haptens are too small to be antigens on their own, but after they bind to something else in your body, then they can trigger immune response. Okay, so remember haptens. So that's just a few words that I want you to be aware of. And on this image here, you can see an antigen. You can see those antigenic determinants. Okay, that's the same thing as the epitope, and that's on that antigen, and that's where your antibodies will be binding to. Okay, and then antigen binding sites, these are things that are on the antibody. That's the part that fits onto that antigenic determinant. Okay, so let's talk about structure of the lymphocytes, and we can see that right here on this image. So there are numerous receptor complexes that are found on each of the lymphocytes. So your whether it's a T lymphocyte or B lymphocyte, they're going to have a lot of these receptor complexes. And on the T lymphocyte, the T lymphocyte receptors are called the TCRs, or T cell receptors. And then on the B lymphocyte, there are BCRs, or B cell receptors. Okay. On the T lymphocytes, you see that there are also some co-receptors there. The CD4 and CD8 proteins, they're also known as co-receptors. Okay. And these are going to help the lymphocyte kind of anchor onto that antigen. So if the T lymphocyte finds an antigen that it can bind to, the co-receptor or the CD4 or CD8 protein will kind of help that cell hold on to it. Okay, so those are your co-receptors there. B cells don't have co-receptors, and the BCRs look like antibodies. Notice that you see that typical Y shape, and that looks like an antibody. So the way that these interact with each other, okay, the way that the cell interacts with an antigen depends on what kind of cell it is. So if it is a T lymphocyte, T lymphocytes, even though they have this co-receptor and all that, they are actually unable to recognize a foreign antigen or a pathogen on their own. They have to have it presented to them. B lymphocytes, they are able to, attach to an antigen, and they know that, okay, this is not okay, and we need to start an immune response against this. But T lymphocytes can't do that. T lymphocytes can be standing next to a bacterium, and it will wish it a good day, and off they go. T lymphocyte wouldn't know. So T lymphocytes have to have this antigen presented to them. So another cell has to process that antigen first and then tell the T lymphocyte, okay, T lymphocyte, this is the antigen. Now start the immune response. And so that's kind of how that works. So we call this process of presenting the antigen presentation. So it's not very weird, okay? It's literally antigen presentation. And this antigen presentation is going to require a couple of things. It's going to require a molecule that is going to hold that antigen. And then it's also going to require a cell that is going to hold that molecule with that antigen and show it to the T lymphocyte, okay? So we're going to need antigen presenting cell and this molecule. This molecule is known as MHC molecule, or major histocompatibility complex molecule, okay? And there are two types for MHCs, okay? There's a MHC class 1 and MHC class 2 molecule, okay? All nucleated cells in your body will have MHC class 1 molecule, okay? And remember, MHC is really just the holder. It just holds that antigen so it can show it to the T lymphocyte. Okay, so all nucleated cells have MHC class 1 molecule. Then there are specialized cells that have other purposes in their lifespan, okay, but they also have this special purpose, which is known as the antigen presentation. And these special type of cells, these antigen presenting cells, will also have MHC class 2 molecule, okay? So all nucleated cells, including the antigen presenting cells, have MHC1 molecule. And the antigen presenting cells also have this special type of MHC, which is MHC class 2. Now, what are our antigen presenting cells? There are 3 of them, and you need to be able to list them, and you need to know which ones they are. So we have 3 antigen presenting cells. B cells, B lymphocytes are one of them, macrophages, and dendritic cells. So if you don't have that in your notes yet, write it down somewhere, okay? Antigen presenting cells could be B cells, macrophages, or dendritic cells, okay? These 3 can act as presenters of this antigen to the T lymphocyte. All right, so let's look at these fun pictures now. Sometimes people don't like them because there's it looks like there's a lot of stuff going on, but it really isn't whenever we focus on what's going on in here. So here we're looking at a regular cell, regular body cell. It's not antigen present, it's just a regular cell that's doing its thing, whatever its function is. One of the normal things that the cell does is formation of these MHC class 1 molecules and these self-antigens, okay? That's one of the normal things that a cell does. And the purpose of this is to show your immune system that the cell is okay, it's healthy, and please don't kill me, okay? That's the whole purpose of this process. So this is just one normal function of every normal cell, every nucleated cell. And so what we see here is that in the cytoplasm, you're going to have some peptide fragments formed. These are going to act as self-antigens. They're going to be little, almost like a little ID tag that tells your immune system cells that they should not attack the cell. MHC class 1 is made in the rough endoplasmic reticulum. And in the rough endoplasmic reticulum, the MHC class 1 molecule and the self-antigen join together. So now you have this MHC/antigen complex, okay? You have the molecule and it's holding on to that self-antigen. We then send it to Golgi. Golgi is going to package it, modify it, do some final adjustment on it, not going into details about that. And then once it passes through Golgi, the MHC molecule with the antigen goes off towards the cell membrane. When it reaches the cell membrane, it's going to fuse with the cell membrane with the antigen sticking to the outside, as you can see on this image right here, right? So, so right here, okay, that's what I'm looking at. You see those self-antigens pointing on to the outside of the cell membrane. So if an immune system cell came by, it sees those self-antigens, it knows that this cell is okay, and it's going to leave it alone, and off it goes, okay? Scanning and checking the other cells in the area, okay? So that's what happens in a normal healthy cell. However, sometimes our healthy cells get infected. They get attacked by a virus or a bacterium or something. And so this image actually shows you what would happen if it's a virus, but it could be pretty similar, you know, for bacteria as well. But viruses are very, very well known for entering cells and causing harm, causing damage. So let's say a virus infects our normal cell that was just minding its business and doing its thing. Now a virus attacks it. So what happens if a virus attacks this normal cell is the cell will try to protect itself, and it will eventually break down the proteins of the virus. And those pieces of that protein from the virus are going to now become antigens on those MHC class 1 molecules. So you still are probably going to have some self-antigens there, but now you're also using these viral particles, viral pieces as antigens. So now you can see what's sticking to the outside. Now we have, we have the foreign antigens sticking to the outside. So guess what happens if an immune cell comes along and it sees this? It's gonna be like, yeah, you're not supposed to be here. Something's not right. And that immune cell will initiate an immune response against this. So, by the way, if a cell is infected, there's no cure for it. There's no hope for it. If a cell has gotten infected, it needs to be destroyed. That's the only thing that we do. So basically that cell will be destroyed somehow, okay? How it will be destroyed depends on what cell first saw that something is not right, okay? So if the cell that's going around and recognizes that, hey, there's a foreign antigen on this cell is sick. If it's a macrophage that recognized it, macrophage will go ahead and destroy it. If it's a cytotoxic T lymphocyte, it will go ahead and destroy it. It's kind of in the name of that lymphocyte, right? Cytotoxic, cyto for cell, toxic for toxic. So that's kind of how that goes. If it's a helper T lymphocyte, it's going to actually not do anything because helper T lymphocytes don't really, can't really interact with that. So anyway, so let's look at our antigen presenting cells here, okay? So this third picture shows you what happens inside of an antigen presenting cell, okay? When antigen presenting cell engulfs a pathogen, such as in this case, bacterium, as it's on the picture, it's not getting infected. So in the previous picture, that cell was not an antigen presenting cell and it got infected, it got attacked and it needs to die. Antigen presenting cells, they engulf things. They are phagocytic cells. They are going to engulf the pathogen and destroy it. They're not getting infected in the process, okay? So a dendritic cell or a macrophage or a B lymphocyte will engulf the pathogen and break it down. In the process of breakdown, you're going to see that the MHC class 2 molecules will start picking up those pieces of that broken down pathogen. Now, in this case, this doesn't happen in the rough endoplasmic reticulum like we saw in the MHC1 case. These guys don't really match up until after the MHC class 2 is already out of Golgi. But that's, I mean, it's a very minor detail there. Okay, but that's what that's where they meet. And then the foreign antigen is, once again, it's expressed to the outside. Now, this is going to be a stimulus or kind of like a calling for your T lymphocyte, especially for your helper T lymphocyte. Okay, your helper T lymphocyte will detect this on the antigen presenting cell, and it will get activated. And when that helper T lymphocyte gets activated, it starts multiplying itself. And all those copies, new copies of that original helper T lymphocyte, are now going to have a memory of that antigen. So it's like they have seen it on their own, even though they really didn't. And so these multiplying helper T lymphocytes will continue releasing these chemicals that are going to activate other immune cells. That's including cytotoxic T lymphocytes and B lymphocytes as well. So that's that. But for that, I have another image. So we'll go over that. Okay, in this image, you can see the dendritic cell presenting the antigen to the T lymphocyte. If we went by the color here, then this would be cytotoxic T lymphocyte. They're using the green for cytotoxic. But it could either be cytotoxic T lymphocyte or it could be helper T lymphocyte. The MHC class 1 molecule on a regular cell will interact with the cytotoxic T lymphocyte, okay? An MHC class 2 molecule on the antigen presenting cell will interact with the helper T lymphocyte. Okay, so what I need you to make sure that you know what goes with what is the MHC class 1 molecule interacts with CD8 co-receptor of cytotoxic T lymphocyte. Okay, again, MHC class 1 molecule interacts with CD8 co-receptor on the cytotoxic T lymphocyte. MHC class 2 molecule is going to interact with CD4 co-receptor on a helper T lymphocyte. Okay, so what you really want to focus on is this part right here, because the one in the part A, it's kind of messy, and you might look at that dendritic cell as, oh, that's an antigen presenting cell, which it is. But here, this is where it's really summarized here nicely for you. CD8 is cytotoxic T cell. And CD4, this is helper T cell. Okay. The way I remember that cytotoxic T cells have CD8 co-receptor is that the word cytotoxic has 2 O's. And if you put 2 O's next to each other and you turn it sideways, it's an 8. I know that's a stretch, but hey, it works for me. And then the helper T cell, I remember that the helper T cell has CD4. The first letter in the word helper is lowercase h. If you turn it upside down, it kind of could look like a 4. So that's how I remember which one goes with which. And then the MHC molecules, you just kind of have to remember which one goes with what. But remember, the MHC class 1 molecule, this is usually going to involve a regular non-antigen presenting cell, a regular cell that got infected, and if it's infected, it needs to die. Well, who's good at killing? Cytotoxic T cells. So cytotoxic T cells would be the ones that interact with that. And the MHC class 2 molecules, these are found on your antigen presenting cells. And so you're going to show this to the regulator of the immune response, which would be your helper T cell. Okay. So that's how those go. We're now going to look at thymus and formation and selection of T cells. So this is kind of just an overview of how the T cells are formed, or where, red bone marrow, right? The T lymphocyte, not T lymphocytes, but the pre-T lymphocytes are going to enter the bloodstream, but they're not going to be doing anything yet. They're going to be traveling towards thymus. Once they reach the thymus, they're going to begin differentiating, okay? So I think this is like kind of showing you a lot of different cells, but we're just going to be talking about the T cells. All right, so we're in the thymus. Remember, thymus is a bilobed organ and it's internally divided into lobules. And each lobule of a thymus has a cortex and a medulla. And so when these pre-T lymphocytes enter the thymus, they're going to have to go through the cortex of the lobule first. Okay. In the cortex of the lobule, what you're going to see is a bunch of cells that are going to have antigens on them that are going to act like foreign antigens. They're not really foreign antigens. They're just kind of like foreign antigens. They're not self-antigens, okay? They don't have the same structure as the self-antigens that you don't want your immune system to destroy, okay? So in the cortex, you have these antigen-presenting cells, and they're presenting these, kind of think about them as practice foreign antigens, okay? And so these pre-T lymphocytes come to this area, and now what they're trying to do is they're going to try to find a match, okay? Because well, I guess I can tell you now, but we're going to mention that later on as well. There are a lot of different antigens, and not every T lymphocyte would be able to match up to everything, okay? So that match is actually pretty specific, and when your body encounters some kind of a pathogen or some kind of antigen for the very first time, you actually only have a few cells somewhere in your body that are capable of matching to it and recognizing that this is something that's not supposed to be in your body. Okay, so here are these pre-T lymphocytes, and they're trying to find something that they could make a match with. Okay, so their little receptors are trying to find a match with this practice antigen. Okay, so they're trying and going from cell to cell, trying to find that match. If they do not find a match, if they're not able to match with anything, then these cells will be stimulated to die. Okay, they die by apoptosis, and that's that. They're done. Actually, 95% of cells that enter thymus are going to die in this first phase. Okay, so most of them don't make a match with anything. And you don't need cells like that. If they're not able to match with a foreign antigen, well, what do you need them for then? Okay, they're bad, so we destroy them. Okay, so only 5% survive that first part. Out of these 5% that survive that first part, some of them are going to be able to match to something that's on MHC class 1 molecule. If you match with MHC class 1 molecule, that means that you would become a cytotoxic T lymphocyte, because MHC class 1 is normally on normal cells that got infected. Okay, so if you're a cell that is making a match with something on MHC class 1 molecule, you're going to become cytotoxic T lymphocyte later. Okay, if you make a match with MHC class 2 molecule, then you're going to become a helper T lymphocyte. Okay, because helper T lymphocytes interact with MHC class 2 molecules. So these pre-T lymphocytes that enter the thymus, they actually have both co-receptors initially. And it's this first step that determines what they will become. It's determined by what are they able to match to, okay? So if you made a match with MHC1 molecule, then your CD4 co-receptors are going to die. If you made a match with MHC class 2 molecule, then your CD8 co-receptors are going to be removed, okay? So you can see that this is known as the positive selection. This first step is positive selection, okay? If that cell binds MHC molecule, then it survives. If it does not bind the MHC molecule, it dies. Okay. And once again, about 95% of cells of these pre-T lymphocytes do not pass this first stage. Okay. So only about 5% move on to the next stage. The next phase happens at the kind of at the border of the, it's not all the way completely in the medulla, but it's in the border of cortex and medulla of each lobule. And what happens in the next phase is now we're going to see if those T lymphocytes that pass the first selection, if they are really for real, or if they're just matching and binding to everything out there. Because if they're binding to everything out there, you don't want them either, because that would mean that they're binding your self antigens. That's bad. Okay. So in the negative selection, you do not want any binding to happen because these now thymocytes are presented with self antigens. You don't want your immune system cells to attack your own self antigens, to attack your own cells. Therefore, it's important that in the second phase, there's no matching, okay? I want you to rewrite this the way that this is written here. I don't know why they use this terminology. Get rid of the word recognizes and replace it by binds. Because using the word recognizes is kind of, it could mean two different things to people. So write down binds, okay? If it says recognizes, that some people would say, well, it recognized its self-antigen, so it didn't bind. Or, well, it recognized it, so it did bind. So which is it? Okay, it's binds. Okay, if in the negative selection, if it binds self-antigen, we gotta destroy it. Okay, we don't want that one. If it does not bind the self-antigen, then it survives. Okay, more than I want to say 2/3 of the smaller group that's now in the negative selection will not pass this. So overall, you have about 2% of the cells that enter the thymus survive and pass both selections. Okay, the first one is much more strict. Okay, and then it's on this, it says, then you have the selective loss of CD4 or CD8. Remember, it is determined by whatever that match was in that first phase. Okay, so if the match was with with MHC1, then it's going to become cytotoxic T lymphocyte. If the match was MHC2 with MHC2, then it's going to retain its CD4 protein. So really be aware of what goes with what. All right, so that's our thymus and our T lymphocytes. Oh, by the way, after these cells pass both of these selections, they're now going to become what we call naive T lymphocytes. Okay, naive T lymphocytes mean that they're functional, they can do their job, but they just haven't seen the real action yet. They haven't seen the real pathogen, so they're naive. Is what we say. All right. So hopefully that made sense. It really, as long as you go over that flow chart, a few times, then you should be good to go. You're basically just kind of memorizing steps of how this happens. The activate or the selection of the T lymphocytes. Make sure that you know what is positive selection, what is negative selection, what's involved in each. So whenever you're studying, really pay attention to those words, to those terms. All right, let's go ahead and look at how we stimulate these cells. What I want you to realize, or what I want you to think about, is that when there's an infection going on, all the cells that are in that area are affected in some way, okay? The tissue cells that are getting infected are going to need to be destroyed. Any immune cells that are in that area are seeing that same infection, okay? So it's not necessarily like one by one doing this process, if that makes sense. But in order to, to put it on paper, we have to kind of start somewhere, right? And so we're gonna begin with the helper T lymphocyte here. I'm gonna begin with helper T lymphocyte, even though cytotoxic is over there on the left. I'm gonna start with the helper T lymphocyte because it is kind of a central cell. It's going to activate other helpers. It's going to activate other cytotoxic T lymphocytes, and it's going to activate B lymphocytes also. So let's go ahead and begin. And I think that's why they actually put it in the middle there. Because if you're looking in your textbook, it's spread over 2 pages, I believe. Okay, so helper T lymphocyte is going to detect or make a make a match with an antigen on an antigen presenting cell. Whenever this match happens, okay, so you got a helper T lymphocyte and you got your APC, antigen presenting cell. When this match happens, that binding causes internal changes in that helper T lymphocyte, and those changes cause the release of a substance that we known as interleukin-2. That's this right here. Okay, so interleukin-2. This interleukin-2 is going to stimulate that helper T lymphocyte, but also if there are any other helper T lymphocytes, naive helper T lymphocytes around, they might be stimulated as well. And it stimulates this helper T lymphocyte to divide. Okay, this helper T lymphocyte now begins to divide, and it creates clones of itself. All of these clones are going to have knowledge of that antigen that this initial first helper T lymphocyte encountered. Okay, so those new clones, it's like you're cloning it with its brain. Okay, it does not have a brain, right? But it's going to act like it has encountered it personally. Okay, so that's how that works. These activated T lymphocytes are also releasing interleukin, and yeah. So part of these helper T lymphocytes are going to help with managing this particular immune response that's happening right now. There's going to be a group of helper T lymphocytes that will become your memory helper T lymphocytes. So should you become exposed to the same pathogen again, you now have thousands and thousands of cells that have the memory of it and can start the immune response much faster. Okay, so there's going to be a group that become memory T lymphocytes. These activated helper T lymphocytes, they're releasing that interleukin-2. The interleukin-2 is also activating cytotoxic T lymphocytes. Cytotoxic T lymphocytes, they can make the match with the antigen on the MHC class 1 molecule. Okay, but they need that stimulation from the helper T lymphocytes to start dividing. Okay, and so as the cytotoxic T lymphocyte made a match at the same time, kind of at the same time as the helper T lymphocyte is making its connection there, the interleukin from the activated helpers is going to tell this cytotoxic T lymphocyte, okay, so now you've seen this this pathogen, so here, divide and send out your army into the area. Okay, that's kind of how that works. Again, there's going to be a group of cytotoxic T lymphocytes that are going to go out there and destroy those infected cells. There's going to be a group of cytotoxic T lymphocytes that are going to serve as memory T lymphocytes. Okay, but it's remember, as the helper T lymphocyte is making the match with that MHC molecule and the antigen in the same area where the infection is going on, the cytotoxic T lymphocytes are also making match with the antigen, with the MHC class 1 molecule. Okay, so that's how that works. So it's not like you first have the helper T lymphocyte make a, detect that antigen, and then you have the cytotoxic. It's they're all in the area together, right? But it's the interleukin from the helper T lymphocyte that is needed for proliferation of these cells, for the reproduction of these cells. So that's our T lymphocytes. Go over that, those figures, and say the steps out loud to yourself. Okay, that I think would be helpful, especially if you're learning the flow of things. I think that really helps. So let's look at the B lymphocytes. And the B lymphocytes are a little different. So this is what happens if the antigen presenting cell is a B lymphocyte. So here in this image, it just said antigen presenting cell. Okay. So we're just going to assume that that's macrophage or dendritic cell. If that antigen presenting cell, it happens to be B lymphocyte, because B lymphocytes are also antigen presenting cells, then the process is a little different. It's still similar, but it's a little different. So the B lymphocyte, when it encounters an antigen, it will bind it with its B lymphocyte receptors. Those will then migrate to the inside of the cell. The cell will engulf those antibodies, with which are the receptors, with the antigen, and then that antigen will be attached to that MHC class II molecule. Helper T lymphocyte comes along and binds to that antigen on the MHC class II molecule. And if the antigen presenting cell is B lymphocyte, helper T lymphocyte will not release interleukin-2, but it will release interleukin-4. So and interleukins are cytokines. So there are these immune system cell molecules. Okay, so but it's interleukin-4 in this case. You might, no, I was going to say CD4 and interleukin-4, but we have CD4 in the other one too. So somehow remember that it's interleukin-4 for the B lymphocytes. And what that does is it stimulates the B lymphocytes. And then the B lymphocytes, some of them will become memory B lymphocytes, and a bunch of others will transform into what we call plasma cells. These plasma cells are going to be like your little mini factories for antibodies. Okay, so they will be mass producing antibodies against that particular antigen. These antibodies will be mostly in plasma, and there will be, well, we'll see what antibodies do here in a moment. But you know one thing that antibodies do, and you know that that's called agglutination. So you already know one of the 6 different things that we will talk about. So that's how these cells are stimulated. Again, whenever you're learning these steps, think about it as kind of a flow chart, and really pay attention to the fact that this image that I have up right now, this is if this antigen presenting cell here that I already circled is B lymphocyte, then we have that different interleukin for B lymphocyte. Okay. And B lymphocytes, once they're activated, they're not like releasing anything, unlike the helper T lymphocyte. So that's that. I do want you to be able to tell me these terms right there. So what is the effector response of helper T lymphocyte? You would say the release of cytokines, and this is going to regulate other cells. The effector response of cytotoxic T lymphocytes is inducing apoptosis in an infected cell. Okay, that's what they do. That's effector response means what do they do? Effector response of B lymphocyte is production of antibodies ultimately. Okay, that's effector response of humoral immunity as well. Antibody, this is a structure of the antibody. Be able to label it. I will make you label it. So pen and paper and draw it and label it, check yourself and do it again. Okay. So just make sure that you're able to label this. There are, we can divide the antibody in from several different kind of angles. You have arm region versus the stem region. There is a heavy chain, light chain. You have constant region, you have variable region. So just know which one is which there. Don't mix them up. There's a hinge region, that's the bend in that antibody. It's held together by disulfide bonds, but the antibody is a protein. So normally proteins their shapes are held together by disulfide bonds. So that's nothing really special there. And then at the end of the variable region is where you have that antigen binding site. The variable region is what is different between different antibodies, and that's why it's called variable region. And each is going to be able to bind to a different antigen. So this is a very simplified version of 3 different antigens and what that would look like. But I mean, of course, they're not little spheres and little triangles. They have slightly different shapes, but hopefully it gets the point across. Okay, so let's talk about what these antibodies do. And when we talk about the function of antibodies, we can look at the area of the antibody that's involved in that function. So here you see that we're at the top, we're looking at what is the function of the antibody and its antigen binding site. Okay, so this is involving antigen binding site. So if the antigen binding site is involved, then the antibody can cause neutralization. And neutralization is where the antibodies will bind to the antigen, okay, to all those epitopes, those binding sites there, and they will physically cover it. Okay, they will physically cover that, in this case, virus on this image here. Okay, they will physically cover it, and so that virus cannot cause harm because it's covered in this layer of antibodies. Okay, so that's called neutralization. Agglutination, you already know agglutination is when antibodies cross-link cells. The important word here is it cross-links cells. Okay, so cells, you would have an antibody cell, antibody cell, antibody cell, and we go like that. So cells are being cross-linked, and it results in the formation of a clump, okay, or agglutinate. And then there's a precipitation. This is a third part, and precipitation looks very much like agglutination. The difference between the two, between the agglutination and precipitation, is that in precipitation, the antibodies are cross-linking soluble molecules like toxins or stuff like that. That's normally dissolved, but if you mix the antibodies with it, it will cause a fallout of the solution. It will cause a clump. Okay, so it's agglutination and precipitation are almost the same. The difference is in what is being cross-linked. Agglutination, you're cross-linking cells. Remember that, you already know that word from blood, right? When we're talking about agglutination. Precipitation is when soluble particles are cross-linked, okay? And result is clump in both in both cases. So that's what the antigen binding sites of antibody can do. But we also have that FC, the stem region of the antibody. So let's see what that does. The FC region can serve 3 functions. They kind of overlap for me, I think. I kind of think that they're really, really related to each other. So the FC region of the antibody, or the stem region of the antibody, can serve for fixation of a complement molecule. And complement is something that we talked about in the innate immunity. Okay, complement is has several different functions. It's part of that innate immunity. So if you forgot what complement does, go back and look at what complement can do. Okay, so one of the functions of complement is actually opsonization. But the FC region, even without complement, can cause opsonization. Opsonization is where you make the you attract immune cells like phagocytes. So like macrophages or even dendritic cells are phagocytes. So the exposed stem regions kind of call for that macrophage to come over and destroy the pathogen. And then the third thing that the stem, the FC region can do is activate natural killer cells. And natural killer cells, as their name suggests, are going to be good at killing things. They're not the same as cytotoxic cells, but kind of similar in the way of how they kill the cell. So natural killer cells are going to be attracted to these exposed Fc regions of the antibody. When they're going to anchor, ultimately, they're going to attach to those ends. When that anchoring happens, that causes natural killer cell to release perforin and granzymes. Perforin will perforate the cell membrane of the infected cell, and the granzymes will go inside and they will stimulate the cell death from the inside. Okay, so the exposed Fc region are complement, fixation, opsonization, and activation of natural killer cells. When you're learning things that are kind of classified into categories like this, what I would suggest doing first is memorize what 3 things go with the antigen binding site, and then memorize what 3 things go with the Fc region. Once you know what belongs where, then focus on what is what, okay? So be able to list it first. That's like a first step, and then go deeper and look at what does that mean, okay? So don't lose the sight of the big picture. This table shows you the different types of immunoglobulins, or different types of kind of antibodies almost. And so you can see that there's 3 of them that are monomers, so it's just a single antibody, and IgA is a dimer, so you got 2 of them attached together, and then IgM is your pentamer. Penta means 5. And so what I would like you to know from this section is I would like you to know the unique characteristics of each. You don't need to memorize the half-life. You don't need to really remember the actions with using that terminology. I think it's easier for you to remember what like specifically where they are found, or specifically what they are doing, which is listed under those unique characteristics. It's easier for you to remember that IgG is the one that crosses placenta, compared to if you were to memorize, oh, it does neutralization, agglutination, precipitation, and all those. So just memorize the unique characteristics and maybe primary locations, but you don't have to focus on the actions and the half-life. So that's that from this table. Slowly getting towards the end, right? Okay, that's just kind of the integrate figures where it puts everything together for you. Okay, the last thing that we're going to talk about is immunologic memory and how we measure it, and then active and passive immunity, and then you're good to go. So the immunologic memory, I've kind of already hinted that, like, initially we have very few lymphocytes that are able to recognize a pathogen, and then the second exposure you would have more, because you build that memory, not immunity. Well, you build immunity and you also build memory. So so we can talk about the first exposure and then subsequent exposures to something. The first exposure, upon first exposure, you only have those few lymphocytes that are able to recognize an antigen. So this is going to take a while. The first meeting of the lymphocyte and the antigen is known as the antigen challenged, and that's what is going to create those memory cells. You can see that it takes several days for the antibodies to even start being formed. So there's that lag phase, because remember, first we go through that innate immunity first, so even that takes still several days. And then once your adaptive immunity is activated, now you're creating those cells, and now you're creating those antibodies, after everything is activated. So it takes several days for antibodies to start being formed. And then the level spikes up, and then it kind of drops a bit. However, if you're exposed to the same pathogen again, what's going to happen is now you have thousands of memory cells, so that lag phase is much shorter. You're not lagging as much. You have thousands of cells all over your body that are able to recognize that antigen. And so the response is going to be much, much quicker. The pathogen is often eliminated before you even know that you were exposed to something. So you might not even know that your immune system was working on eliminating something, because your cells, you have so many of those cells and they're so quick, they just destroy that pathogen. I won't even give it a chance. Okay. So so often you don't have any symptoms. And yeah, that's that for immunologic memory. We measure immunologic memory by measuring antibody titer. Antibody titer is basically a measure of of the antibody levels in your blood. So you would have them, kind of fairly high, at a certain point after you were ill. But if you got exposed to it several different times down the line, you might not have known that. And so your titer might be quite high, even though you think that you only got sick with it once, right? So that's how, so we can measure it. So antibody titer measures the levels of antibodies in your blood. And every subsequent exposure spikes that antibody titer higher, okay? So the more you're exposed to something, the higher your antibody levels are. And so two people that maybe got, I don't know, some sickness at the same time, 10 years later, they might not have the same antibody titer because one of them might have been reinfected by it more than the other. So that's that. They're actually, the reason that the fact that the second or subsequent exposure to something you've already been exposed to spikes up your antibody production is why I believe that they're recommending the pertussis vaccine for pregnant women. And I don't remember if they recommended that with my first one, but I know that was a recommendation when I was pregnant with my second one. She's now 6. And so somewhere around like week 30-ish or so, they said, okay, go get the pertussis vaccine so that your body produces these antibodies against it. Pertussis is a whooping cough. And antibodies can cross placenta as well as breast milk. So that was the idea behind that, is that there would be some protection for the newborn. Don't really know what the results of that are. Like how effective it is, but pertussis is the most deadly for children under 2 months, and infants cannot get the vaccine until they're 2 months. So during that deadliest part of that contracting the pertussis, they cannot be vaccinated. So I guess that's why they were recommending to women to get vaccinated, so we can pass on the antibodies to the baby. Again, I have not seen the studies of how effective that actually is, but that's just the idea behind it, like why they're doing it. The last thing is active and passive immunity. So here again, I would recommend maybe draw the flow chart yourself a few times so that you know what belongs where. This is really not that difficult. Just make sure that you are aware of what belongs where. So we can talk about our immunity between being active or passive, and this depends on how your body is getting the antibodies. So is your body making the antibodies itself, or are you getting the antibodies from something else or someone else, not something? So in the active immunity case, your body is the body that produces the antibodies. So your body produces plasma cells, and those plasma cells are actively making antibodies against that pathogen. You can acquire active immunity naturally or artificially, okay? So naturally acquired active immunity is when you get sick. You get sick from something, okay? So somebody sneezes in your face. Well, now they should be having masks, right? We're all wearing masks now. Yeah, somebody coughs at you, somebody sneezes at you, or you touch something and then it enters your body. Somehow you get ill, and then your body goes into the full-blown immune response and you recover. Okay, that's your naturally acquired immunity. Artificially acquired immunity is when you're exposed to the antigen from a vaccine. And there are different kinds of vaccines. Some would be using like parts of the pathogen. So they would take like a virus and break it down, and it just has parts of the viral capsule. And then your body still perceives it as that antigen. It still has those epitopes on it. So your body can fight it off. Sometimes they're using weakened pathogen, like there's some flu vaccines that are using weakened flu virus inside. And so that virus is, it's not active at the body temperature that our body is naturally. So technically flu vaccine cannot give you flu. But anyways, that's that. And yeah, that's artificially acquired, is from a vaccine. Passive immunity is where your body is not making those antibodies itself. So you can actually, there is a naturally acquired passive immunity, and this involves mother and a child. So it's either antibodies passing through the placenta to the fetus, or it's antibodies going from the breast milk to the infant. Okay, that's naturally acquired passive immunity. The baby is not making the antibodies, but it's receiving those antibodies from the mother. So we consider that as the naturally acquired passive immunity. Artificially acquired passive immunity is where you transfer serum that has the antibody to another person or an animal. So I know that was, they were doing this when we had the Ebola outbreak, I guess, a few years back. Like they were using that doctor's plasma somehow to, try to treat other people actually. But the vaccine against tetanus and botulism is made or is a like a transfer of serum. Snake bites also. Okay, so you can treat snake bites with the serum somehow like this, with the antibodies against that toxin. So yeah, so that's that. So make sure that you know what belongs where. I will have some examples on the exam, and you'll have to tell me whether it's active or passive, and then whether it's natural or artificially acquired. So you'll have to classify some scenarios there, which I think I don't think I have anything new. I pretty much described them right now. So anyway, so that's that. Okay, any questions about this? I know second part of immune system tends to be a little intense. There's a lot of stuff. No, we didn't go too far, over an hour. All right, good. Are you just ready to go process it now? All right, well, good luck. If you have any questions, you can always email me, and I'll see you on Wednesday.
Thank you.